Hello students, welcome to Distinction Learning Platform. In this video, we are looking at enzymes. But before we begin our lessons, make sure that you subscribe, like the video, as well as don't forget to comment in the comment section. Alright, let us begin. So in this video, we are looking at the factors that affect the rate of enzyme activity. And if you didn't watch the enzyme video where we talked about the classification of enzymes, make sure that you watch that video. Alright, now what are the factors that affects the enzyme activity? Remember that what is an enzyme? An enzyme, this is the biological catalyst that speeds up the rate of chemical reaction without itself being changed. You need to remember about that, right? Now, the thing that you can ask yourself is that these enzymes, since these are biological catalysts, basically these are considered as living things. So if they are living things, they can be affected by various things. What do I mean affected? It means that the way they work, is either they increase their work or they decrease their work. Okay, so look at that. So let us look at those various factors that can affect how these enzymes work. Okay, so now what are those factors? So we're talking about factors that affect enzyme activities. Okay, factors that affect the enzyme activities. What is the first factor? The first factor is what we call pH. Okay, so let's talk about pH. The first factor is what we call pH. Okay, which is pH stands for what? Potential of hydrogen or power of hydrogen. If you didn't know, make sure that you know. Power of hydrogen or potential of hydrogen. Okay, and the second factor that you need to remember is temperature. Okay, temperature is a very important component that will affect how enzymes are going to be working. Alright, then the other components or factor, remember that will be now concentration. Okay, remember that the higher the concentration, the higher the rate of enzyme activity. That's something that we always talk about. Alright, so that is also concentration. And there are other various factors that can affect this. Okay, like the cofactors that we'll talk about. Cofactors. Okay, and number 5, inhibitors. Okay, we'll talk about that. Okay, so now let us start with the pH. Okay, how does the pH affect how enzyme work or enzyme activity? So we need to remember about that. pH, potential of hydrogen. So let us describe about the pH. The pH. Okay, power of hydrogen. So let us write that power of hydrogen or potential of hydrogen. Power of hydrogen. Okay, and we need to remember that if you want to understand about the pH, we need to remember the pH scale. Okay, so let us write the pH scale so that we can remember if the solution is acidic, normal, or alkaline. Okay, or neutral, I mean. So let us write about the pH scale. Okay, remember that the pH scale, well, is going to look like this. So the pH scale is going to look like this. Remember that it will start from 0, 1, 2, 3, 4, 5, 6, then 7. We're going to change now the color. Then 8, 9, 10, 11, 12, 13, up to 14. Okay, then this 7 I'm going to use red so that we see the difference. Okay, this is what we call the pH scale. Okay, this is what we call pH scale. pH scale. Okay, so now let us understand this one, because minus this we can't understand how pH can affect the rate of enzyme activity. So this pH now is going to have these numbers from 6 to 0 going like this, okay? What do we call these numbers? So what do we call this region? This region is what we call the acidic pH, okay? So what do we call this part? Starting from where? Starting from 6 up to 0, we call it acidic pH. Okay, you need to remember this. Acidic pH is going to contain numbers or lower numbers than 7. Starting from 6, then up to 0, then we are going to call it acidic pH. Now, what about this 7? This 7 is what we call neutral pH. So this 7 is what we call the neutral pH. I'm going to use red for this one because it's a special pH. Neutral pH. So neutral, something that is neutral is not that dangerous to our body. A good example that I can give, a substance with a neutral pH is sodium chloride, which also known as table salt. Table salt, that's why we are able to put it in different layers, because it contains neutral pH. It is not dangerous to our body. Something which is neutral, it is not that dangerous to our body. Okay? Now, what about this? So, what about this starting from 8 to 14? What type of pH is this one? We call it alkaline pH. The reason why it's called alkaline pH, because as you can see, the numbers are now increasing or are more than 7 up to 14. Alright? That's what we call alkaline pH. Now, the question that you can ask is that, how are these pH values going to help us understand enzyme activities, or how they are going to decrease or increase the rate of enzyme activity, or how enzymes are going to be affected? Okay, I'm glad you asked something about that. All right, now if you look at the body, okay, if you look at the body, where can you find the acidic pH and where you can find the alkaline pH? You need to know. So in the body, where you can find the acidic pH, or where you can find the acid, is where? In the stomach, alright? So the region in the body where you can find this acidic pH, will understand how this is going to be related with the enzyme activity. So let us just try to understand where you can find this type of pH in our body. Okay, so where you can find this type of pH in our body is what we call the stomach. Okay, this is where you can find the acidic pH, because in the stomach, that's where you can find the hydrochloric acid. Okay, the alkaline pH, you can find it where? You can find it in different regions of the body, like the mouth. Okay, you can find it in the intestines. Okay, intestines, large intestine, small intestine. That's why we can find the what? The alkaline pH. Alright? So at least you know where you can find this type of pH. The neutral pH, quite alright. I don't think there's a region for neutral pH, right? Now, if you have a question like this now, so let us look at now how these are going to affect the enzyme activity. If you look at the stomach is going to have different enzymes that will be working in the stomach. Okay, so let us get a good example of that enzyme that's going to be working well in the stomach. Like what? Let us get one good example of an enzyme that's going to work in the stomach. Is what we call pepsin. So let us just get pepsin. Okay, pepsin is a good example that works well in the stomach. Okay, now the problem is this: if you get this pepsin and transfer it into alkaline pH, like the mouth or the intestine, what's going to happen to this pepsin? Again, what I'm simply saying is that if you get this pepsin and you transfer it into another region, because pepsin, it is working well in the acidic pH, where? In the stomach. But by all means, you get this pepsin and you transfer it into what? Maybe in the intestine or in the mouth, where there is what? Alkaline pH. What's going to happen to this pepsin. This pepsin will be affected. It's going to be what? The way it is working is going to be decreased. Okay? Technically, we're going to say inactivated. So this one is going to be what? Inactivated. If it is transferred to a region where it is not comfortable, it's going to be inactivated. But in the region where it is comfortable, like in the stomach, it's going to be what? Active, which is it's going to be activated. So that's the main thing that you need to understand. So pepsin works best in the stomach. We understand about the digestive system. Okay, works well in the stomach and it's going to be activated, meaning that it's going to be active, trying to digest the proteins there. But once it is transferred in the like intestine mouth, it's going to be inactivated because it doesn't like alkaline pH. Alkaline pH, which is starting from 80 to 14. That's how it's going to affect the enzyme activity. Alright? Now, another important concept that we need to understand here is this one. If you have a graph like this, okay, if you have a graph like this, okay, on a graph like this, then this side it is the rate of enzyme activity. So this side it is saying rate of enzyme activity, rate of enzyme activity. Okay, this side on the y-axis there is rate of enzyme activity, and this side there is what? pH. Okay, pH. Remember that pH is starting from 0. Then here maybe this is 2. Maybe here this is 4. Maybe this is maybe this is, let me just say 5. Okay, and maybe this is 7. Okay, this is 8. This is 10. This is 13. Then this is 14, like that. Okay, we have this type of what? Graph representation about the pH, and this is A. This is region A and this is region B. Okay, and they say they tell you this is reaction P, and this is reaction M. Okay, now they can ask you questions like this. They can ask you a question like, identify region A. Where can you find in the board this region A? This is region A. As you can see, this is region A. Where can you find in the board this region A? Where can you find it? You can find region A where, as you can see, this one is telling us to say this region A is within the what? Is within the acidic pH. Okay? Remember that what I said, the acidic pH, which region can you find is what? The stomach. So the moment they ask you to say where can you find region A, what can you say? You can say you can find it within the what? The stomach, because that's where acidic pH can be found. Because if you look at the pH here, this is 0.25, then somewhere 70, but the majority of the numbers are within the what? The acidity range. That's why we're saying A is the stomach. Then what about B? B is within like the alkaline region. If they ask you to identify region B, what can you say? You can say it is the intestine or the mouth. Okay? Then they can ask you about the reactions. So they can ask you identify the reactions, identify reaction P in which region A, in which it can take place in what? Region A. So the reaction, they're just talking about what type of reaction that can happen in region A. The reaction that can happen in region A, basically, that is the digestion of proteins in region A. For B, it can be a lot digestion of starch, digestion of what? Other molecules. So in simpler terms, this is how they can bring a question under what? pH for enzyme activity. Alright? So now let us proceed to another important concept. Temperature. So let us talk about temperature. Okay, so if you look at temperature, Remember that temperature is the measure of how cold or hot a substance is. That's what we call temperature. Now, if you look at the temperature within our body, the optimum temperature within our body is what we call, is around what we call 37 degrees Celsius. That's the normal body temperature that we need to remember. Okay? Now, the question that we can ask is that, how do temperature affect the rate of enzyme activity? Temperature is going to affect the rate of enzyme activity is by, if you increase the temperature, you're going to increase the rate of enzyme activity. But if you increase the temperature more than 37 degrees Celsius, you're going to decrease the rate of enzyme activity. So what do I mean? I mean like this. I'm going to be given a graph like this. Okay, this side is for temperature. Okay, and this side is for rate of enzyme activity, for y-axis, this side. Rate of enzyme activity. Okay, this side. And this is for the values for temperature. Okay, and you're going to have here maybe 37, here you're going to have 33, maybe you're going to have 22, up to maybe 0 here. Okay, then here you're going to have maybe 38, okay, 39, maybe somewhere here. Okay, you are going to have 40 somewhere there. Okay, so this is the graph for temperature. Now, the question that they can ask you is that, okay, if you look at this, what did I say? From 0, as you're increasing the temperature, okay, if you look at it, as you're increasing, if I'm going like this, mean that I'm increasing the temperature here. So as I'm increasing the temperature, I'm also increasing the rate of what? Enzyme activity. But up until I reach this what? 37. This 37 is what we call optimum temperature. What is optimum temperature? Optimum temperature is the type of temperature where enzymes work the best. So optimum temperature, I'm saying this is around what? 37, and the definition is that this is the temperature where enzymes work the best. So enzymes work the best. This is what we call optimum what? Temperature. Enzymes work the best at 37 degrees Celsius because this is what we call the optimum temperature. Now, what we're simply saying is that the more I'm increasing the temperature, also the rate of enzyme activity is what? Increasing. But the moment I hit 37 here, this is 37. The moment I hit this 37, the graph will start what? Decreasing, showing that now there is maximum heat exceeding 37. The enzyme will start being what? Denatured. Okay. So what we are simply saying here, after the 37 degrees Celsius, this graph, this part is telling you that enzymes are being denatured because of high temperature, are being denatured. Okay, enzymes are what? Being denatured. You need to understand. But at 40, as you can see, at 40, that is completely denatured. Okay, we need to understand about that. So after 37, enzymes are being denatured, but at 40 degrees Celsius exactly there, enzymes are now completely denatured because now the graph reaches what? 0. It's coming back to 0. Then we're going to say enzymes are now completely denatured. Denature, this word denature, what does it mean? Being killed. Okay? Enzymes are being killed, but we don't normally use kill in biology, but we use what we call denature. Okay? This is how temperature is going to affect the rate of enzyme activity. Remember, the higher the temperature, the higher the rate of enzyme activity. But if the temperature exceeds 37 degrees Celsius, we're going to have what now? The rate of enzyme activity to start decreasing because the temperature has exceeded 37. Now the rate of enzyme activity is going to be what? Decreasing. Why? Because the enzymes are now being denatured because of what? Extreme high temperature. Okay, that's how enzyme activity is going to affect that's how the temperature is going to affect the enzyme activity. Okay, now let's go to concentration. So let's talk about the concentration. Okay, so concentration is what? So remember that concentration is the amount of substances that are dissolved in a certain solute. That's what we call concentration. Now here, we're going to have two important concentration, because if you talk about concentration, we're talking about the amount of substances in a certain volume or solvent. That's what we call concentration. Now, this concentration, we're going to have the concentration of two important components. We're going to have the concentration of what? Enzymes, and the concentration of what is substrate. Okay, the concentration of enzymes and the concentration of what is substrate. So the concept is very, very simple here. Okay, if you have lot of enzymes, okay, if you have lot of enzymes in a certain region, like let's say in the stomach or the mouth, if you have a lot of enzymes, it's more like you have lot of workers. What's going to happen to the job? Basically, you're going to finish the job faster because you've got what? A lot of workers. Same applies to the enzymes. If you have lot of enzymes, the rate of enzyme activity is going to increase. Okay. What about substrate? If you have a lot of substrate, it also increases the what? The rate of enzyme activity. No wonder they just simply say the higher the concentration, the higher the rate of what? Enzyme activity. Okay, so that's what it means. Now, you need to be understanding the graph of these the enzymes, the concentration of enzymes, the concentration of what? The substrate. Okay, so we need to understand about this. So if you have this, okay, this is the graph. Now the graph of the enzyme substrate is more like this. Okay, it's way different from the one I was talking about under temperature and the one that I was talking about under pH. It looks like this. As you can see, the graph is somehow constant after a certain level. Okay, we need to understand what it means when it reaches this constant part. It doesn't go down, it doesn't go up, but it's more like constant on the same what number. So if this is this part, this is concentration, and this is the rate of enzyme activity. This part on the y-axis, this is the rate of enzyme activity, and on the x-axis, that is where you can find the concentration of either enzymes or substrate. Again, maybe if you are confused with what I mean if I say substrate, so substrate and enzymes. So if you have a substrate, so if this is an enzyme and this is a substrate. So remember that a substrate, it is a substance where enzymes are going to act upon. Okay, so if this is an enzyme and this is a substrate, so this enzyme, they always act on this substrate. Okay, substrate, good example that I can give is maybe the food nutrients. Okay, the nutrients that we take in, those are what we call substrate, because on those nutrients, that's where enzymes are going to start acting upon. Like what? Proteins, lipids, okay, carbohydrates, those are what we call substrates. Alright? So either way, the graph of the concentration of enzyme substrate against the what? The rate of enzyme activity looks like this. Okay? But here what we just have to understand here is this part. Okay? Why does it remain constant? Okay? Because if you remember, the graph for temperature, it was like this. It was coming down because the graph was exceeding what? 37 degrees Celsius. Okay? If you also remember the graph for what? pH, it was more like also decreasing like that. Okay? Because also we are changing the pH values from maybe alkaline to acidic. Maybe from acidic to alkaline, it's changing. But this one remains constant. Why is it like that? Okay? The reason why it's like this is this. The moment it reaches this constant, this part, okay, what happens? It means that here there are two main reasons. If you are talking about the enzyme, so the reason here is for the enzyme. Okay, if it is constant, if you are on the x-axis here, there is enzyme concentration. Okay, the reason should be this one. Okay, so meaning that we're going to say the enzymes are filled with enzyme active sites. So I'm going to say enzyme active sites are filled in with the substrate. Okay, so what we're simply saying is that if the constant, if the value reaches here, okay, what does it mean? It means that all the enzymes that are there, they are now occupied with what? The substrate. Okay, they are all working. If they are all working, basically there's no need to increase the rate of enzyme activity. It's going to become constant. Why? Because the enzyme active site. Okay, so let's just say active site. Sorry. Enzyme active sites are filled in with a substrate. Simply means that all the enzymes that are there, they are what? Working, okay? They are filled. Because if you remember, if this is an enzyme is going to have different parts. If you remember from the other video, we said the enzyme is going to have this part where this substrate is fitting, is what we call the active site, okay? And this part which is outside is what we call allosteric site. But we're simply saying if this all active site of the all enzymes are filled in with what? The substrate, basically the graph is going to remain constant because all the enzymes are now filled in with what? The substrate. Okay. Same applies with if we had the concentration of what? Substrate. So concentration of substrate, if the graph remains constant, what could be the main reason? The main reason for this one is that we're going to say the substrates are now what? Finished. Substrates are finished. Okay, what does that mean? Meaning that these substrates, they have been what? Digested by what? By these enzymes that we're talking about. Okay, that's why the graph is going to remain constant, because now the substrates that are here, they are now digested by different enzymes. They are done. That's why this graph doesn't have to go up or down. Okay. Now that's about the concentration. Now the last part is talking about the cofactors, which is this one, cofactors and inhibitors. Okay. So cofactors and inhibitors. So let's just say cofactors and inhibitors. So what are cofactors? Cofactors, these are substances that are going to promote enzyme activities. So cofactors again, what are cofactors? Cofactors, these are substances that are going to promote enzyme activity. They are going to allow the enzyme to start working. Those are what we call cofactors. Okay? So here we are saying cofactors, let's just say activates enzymes. Okay, so cofactors are going to activate enzymes. Basically, they are going to promote enzyme activity. Inhibitors, they do opposite function. They are going to inactivate. So the opposite of activate is to inactivate enzymes. So cofactors, they activate enzymes. Inhibitors, they are going to inactivate enzymes. All right. Now, the question that you can ask is that what are the good examples of these cofactors? The good examples of these cofactors, we have a lot of cofactors in our body. We can have vitamin K. We can have vitamin K as one of the cofactors because it helps in the coagulation cascade. Okay. Then we have vitamin B6. Vitamin B6 as well. It's one of the cofactors that we need to understand. Calcium ions. Calcium ions, these are also one of the good cofactors because they also help in different metabolic reactions of our body. So these are good examples of cofactors: vitamin K, vitamin B6, calcium, vitamin A lot of what substance that helps in what activate enzymes. So these are going to help to activate certain enzymes. Now, what about the inhibitors? What could be good examples of inhibitors? Again, inhibitors, there are a lot of inhibitors in our body, but let's just say in simpler terms, let's just say, remember one good example of an inhibitor, we call it poison. So let's just say poison is one good example of what an inhibitor, because the moment you drink poison, basically it's going to inactivate all the enzymes or denature all the enzymes by altering the active site and also allosteric site, so that those enzymes, they can become inactive forever, and definitely the person is going to what be in a dangerous situation. Maybe it can lead to what death. Okay, now this is where we end from under enzymes. Now the factors that affect the enzymes activities, and make sure that you study more. Until next time.